Hello everyone. It gives me great pleasure to come back again within a week actually to talk about part 2 of basics of cancer biology. So today I'll be talking about cancer immunology, but before we get into cancer immunology, it is essential to understand some aspects of immunology itself. It gives a much better understanding of what goes wrong in cancers. And more importantly, now we have a large number of therapeutic measures which manipulate the immune system and thereby are able to achieve better disease control and even cures. So, this is today's presentation, and as ever, I would hope that it is helpful to you all. And I would like to thank all the people who have been kind enough to uh, give some good uh, feedback on the part one. Thank you. So I'm Dr. Raj Kumar. So let's get on with the basics of cancer immunology. So moving on, I have put on two cartoons here. The top one shows the number of COVID deaths which have happened since beginning of the pandemic, and the bottom one is a movie poster on the War of the Worlds. So, what is common between these two pictures? Yes, you're right. There is lack of immunity to the new COVID virus. And that's the reason why we had a large number of deaths here. Secondly, we also find the lack of immunity in the aliens to the microorganisms on the earth resulted in them dying and we becoming victorious. So broadly, I would classify the immune system into innate immunity and adaptive immunity. Innate immunity is what we are born with. So from the time of birth, we have protective mechanisms which can be physical and chemical, like the skin covering, the coverings of the mouth, the GI tract, what we call the mucosal covering, as well as chemicals, which includes different mucins which can protect, as well as also the hydrochloric acid in the stomach which can digest pathogens which try to enter. In addition, we also have a wide range of cellular elements which will also have a role to play, and these include the dendritic cells which we'll talk about later, the macrophages which are phagocytic, different polymorphonuclear leukocytes, basophils, eosinophils, mast cells, endothelial cells, natural killer cells, natural killer T cells, gamma delta T cells, which is a slightly different form. And we also have the humoral innate immunity, which comprises of the complement system, what we call as acute phase reactants, including the C-reactive protein, and then we also have naturally occurring antibodies. So these are usually present in the blood or in the body fluids, serving to protect an individual at the first point of entry of, let's say, a pathogen. In contrast to the innate immunity, adaptive immunity needs to develop over a period of time after birth. So let's presume that we have a virus entering into our system. One of the cell which responds fast is the macrophages, which will take up this pathogen, what we call as a process called as phagocytosis. It engulfs and digests it. The digested pathogen, the different proteins are broken down into small peptides, and these peptides are then exhibited to the T lymphocytes. The naive T lymphocyte becomes activated and it becomes the T helper cell, which will then promote the development of the cytotoxic T cell or the killer cell, which can go and destroy cells which have been infected with this virus. In addition, there is also stimulation of the B lymphocytes, and these will then proliferate and become developed into what we call as plasma cells, and the plasma cells start secreting antibodies, which can again neutralize these viruses. In addition to this pathway, we also have sets of some of the T cells and the B cells which have been sensitized to become memory cells. So the memory cells stay back, and if the same pathogen were to come back again, they trigger off the immune response in a much faster manner and in a much robust manner, leading on to much better protection. So this is again a repeat of what I've just said, just to give you the time scale here. So in innate immunity, this is what exists soon after an infection occurs on up to maybe 12 hours. These are the agents which will start fighting that infection. Whereas the adaptive immunity takes anything from 7 to 14 days or even more. The more later it tends to become more mature and more robust. One of the other cellular component here, which I've shown here, is the T regulatory cells or Tregs. The T regulatory cells is bound to inhibit the activity of the T helper cells, the Th1, Th2, Th17, which are all involved in different immune processes. It also inhibits the cytotoxic T cell by using adenosine, which is derived from the energy source ATP. So ATP is broken down to adenosine monophosphate, and then from that you get the plain adenosine, which is inhibitory to the cytotoxic cell, T cell. It also inhibits the dendritic cell or the antigen presenting cell. So thereby, this regulatory cell controls the immune system. Why is it here? This regulatory cell plays an important role to make sure that the immune system doesn't run amok. If it were to run amok, we end up with reacting against our own self, our own cells, our own tissues, and that results in a condition called as autoimmunity. Autoimmunity can be disastrous, and although there are several therapeutic approaches available, this essentially is multifactorial, and lack of T regulatory cells is one of the features there. The dendritic cell, as I just mentioned, are the most potent antigen presenting cells. And for these antigen presenting cells, they take up, for example, a virus, chew it down, break down into peptides, and these peptides are exhibited through the MHC molecules, which you'll see in little detail little later. This will be presented to the naive T cell, which then becomes activated and then can give rise to the T effector T cells. The effector T cells in turn can activate macrophages which are already carrying or having the viruses within them, so that they promote the destruction of these viruses or the pathogens. In addition, the effector T cells can also activate the B cells to produce antibodies through the maturation of B cells to plasma cells. So this is called the humoral immunity. So what are the components of the immune system? So you have an afferent arm, the effector arm, and the cytokines. So the afferent arm consists of the antigen presenting cells, MHC molecules, and the interaction with the efferent arm. So we'll see each one of these shortly. So as I said a little while earlier, the antigen presenting cells, the dendritic cells are critical. So they activate the resting or the naive T cell, which gets activated and becomes activated T cell, which in turn secrete a wide range of chemicals called as cytokines, which have specific functions, mobilizing more stronger immune response, and also cytolysins, meaning they can actually lyse cells, kill cells. So this is one of the important components of the dendritic cell function. Now, how does an immune cell recognize an infection in a cell? So let's presume we have a cold or virus infection, and let's presume this is our throat cell. So once the virus enters into the throat cell, we have some machinery within the cell which can break down the viral proteins into small fragments called as peptides. These peptides will then be bound by a protein called as MHC complex, MHC class 1 complex. The MHC class 1 complex with this viral peptide will be pushed to the cell membrane so that it can now exhibit that viral peptide antigen to the immune system. So it's essentially a flag. It's actually saying, look, I am infected, please come and destroy me. So this is what will happen. So you have the killer cell now coming in, identifies the viral peptide, and it will then kill this infected cell and tries to prevent the spread of the virus into other cells. Now the major histocompatibility complex or the MHC molecules I mentioned a while ago, these are polymorphic, meaning now one individual has the same kind of patterns. They're highly variable entities, except maybe twins born of the same zygote. Otherwise, there is always bound to be some variations. Now, the MHC molecules can bind the peptides and present them to the T cells. Now, there are different types of MHC molecules. The class 1, which is expressed in all the somatic nucleated cells. In contrast, the class 2 MHC molecules has restricted expression in, let's say, the B cells, the macrophages, that is some of these immune cells, the antigen presenting cells like dendritic cells, follicular dendritic cells, activated T cells, etc. We also have the class 3 MHC complex, which includes some of the components of the complement system, which is a part of the humoral system, and different cytokines, including tumor necrosis factor alpha and heat shock protein. So all of these together constitute the major histocompatibility complex. Now a word about this major histocompatibility complex. So to distinguish between self and non-self, meaning a given individual and something coming in from the foreign source, these are our defense mechanisms. So this is very critical. So as I mentioned earlier, the MHC class 1 has two proteins. One is the alpha protein, and then we also have beta 2 microglobulin. Now, beta 2 microglobulin is present in chromosome 15, whereas all the MHC complex is in chromosome 6. Now, there are 6 loci of MHC class 1, A, B, C, and then E, F, and G. Now, as mentioned earlier, MHC class 1 is expressed in all somatic nucleated cells. Their function is to inform the immune system that they are damaged in the form of either some viral infection or developing a mutagenesis, some cancer-related protein has developed. So all of that goes through this MHC class 1. So as I said, here we have the alpha protein. So it has 3 domains, alpha 1, alpha 2, and alpha 3. And alpha 1 and 2 will have the cleft, and it is here that the peptides will join. And then we also have the beta 2 microglobulin providing support here. This is a peptide from a virus and another viral peptide. So there are two different ones which actually is bound to the MHC1 complex, and this is the cleft. So you're looking at as if the T cell receptor is seeing these peptides, and it is this peptide which will stimulate the T cells. In contrast, the MHC class 2, as I mentioned earlier, is restricted expression in B cells, activated T cells, antigen presenting cells like dendritic cells, macrophages, etc. So this has two chains, alpha chain and a beta chain, which together form a cleft through the alpha 1 and beta 1. So this cleft is where the peptide will be bound to. So what are the molecules involved in the effector cell, target cell, adhesion and antigen recognition. First of all, here is the T cell, which is a cytotoxic T cell. And here is it, let's say, a tumor cell. So the tumor cell is going to express only MHC class 1. So within this tumor cell, let's presume that there are some proteins which are mutated, and these mutated proteins are broken down, and the mutated peptide is now exhibited by the MHC class 1. Now this will be recognized by the T cell receptor, and in addition, because it's CD8, that is a cytotoxic T cell will have a CD8 a protein expression, which also will be critical in improving the signal. Along with this, we have what we call as co-stimulatory molecules. Now the cytotoxic T cell has CD28, and the tumor cell will have co-stimulatory molecules B71 or B72, which are otherwise called as CD80 and CD86. The CD28 combining with either 71 or 72 is very important for the tumor cell lysis to happen, for the signals to actually proceed. In contrast, in some of the tumors, in some of the situations, you find that this co-stimulatory molecules B71 or B72 may be lacking, or instead of CD28, you will have CTLA4 coming into play, which is actually inhibitory, and as a result, the tumor cell can survive. We also have other proteins here which are shown like ICAM. Now these are required for cell adhesion. You need these two cells to come to close opposition so that all these interactions can happen, and that is brought about by this interaction here. So what are the effector arms? So the effector arms we already dealt with: the cytotoxic T cell, the natural killer cell or NK cell, which is part of the innate system, macrophages, again part of innate system, lymphokine activated killer cells, antibody dependent cellular cytotoxicity. And then the soluble or humoral effector system includes antibodies and their complement. I was fascinated as an undergraduate medical student by Burnett's Cancer Immunosurveillance, and it was fascinating to see how our system, our immune system, can help protect us. And this particular presentation I would like to dedicate to Professor Burnett. So what is the anti-tumor immunity? So here, let's presume that there are some cells which have gone astray, the tumor cells. One important concept we need to understand here is that a tumor is not something which has come about from a foreign source. These are cells within our body which has turned malignant. So they are actually derived from our own cells. So that is something which we need to understand. They are not from somewhere from another person or from the plants or animals, etc. So these are cells, the tumor cells, which basically are derived from our own cells, normal cells. Once the tumor develops, you find that the tumor, when it starts developing and rapidly proliferating, some of the cells will die and they will release several tumor associated antigens and proteins. Now these are called as damage associated molecular patterns, or DAMPs. Don't worry about the terminologies. It's just suffice to say that this is like a trigger for the immune system to recognize that something is going wrong. And here we have these molecules which are released from the tumor are taken up by the dendritic cells. The dendritic cells then migrate to the lymph nodes where they can activate other T cells, other antigen presenting cells, and improve the immune response. So you get activated T cells resulting in T helper cells, which in turn can give rise to cytotoxic T cells. It can also stimulate B cells as well. So the T cells, both cytotoxic and T helpers, move out of the bloodstream into the tumor area, wherein they can then go ahead and destroy the tumor. So this is what happens in the destruction process. And the cytotoxic T cells uh, actually cause pores. We'll see that in a short while. One of the ways by which a cancer cell can escape is when it undergoes what we call as a tolerogenic cell death, in contrast to immunogenic cell death. So what is this? So here we have a cancer cell, and let's say that it has been treated with some low dose chemo radiotherapy. This will cause the cancer cell to die, and during the process of death, it releases several of its tumor associated antigens, several products from within the cell, including what we call it the DAMPs, the damage associated molecular pattern proteins. All of these will stimulate the mature antigen presenting cell. The mature antigen presenting cell migrates to the lymph node where it can now activate the T helper cell, which will in turn secrete IL-2 and other cytokines, stimulate the cytotoxic T cell, which can then go and destroy the additional tumor cells. And this it does by secreting a protein called as perforin, which causes pores, or it produces punctures on the tumor cell, causing leakage of the cytoplasm and the material from within the cell, leading to death of the cell. And in that process, there is more immunogenic cell death happening, so more immune system can get boosted. Now this contrasts with the tolerogenic regulated cell death, as you see here. So here, this is an apoptotic cell death, and in apoptosis, the membrane bound bodies prevent any of the release of these materials, the DAMPs, from stimulating the antigen presenting cells. Once this happens, there is no migration of these antigen presenting sensitized mature dendritic cells into the lymph nodes, no activation of T helpers, no activation of cytotoxic T cells, so no killing of the cancer cells. So which means these tolerogenic cell death can actually promote cancer survival. The same one which I mentioned a little while ago, the cartoon, but here it gives you a much better understanding of the different factors which come into play. So I've already talked about immunogenic cell death here and tolerogenic cell death here. Here we have the released products, which include a wide range of compound molecules, as well as which can be stimulatory, which are given in green, and those which are inhibitory are given in red, like IL-10 or IL-4 or IL-13. So you find that some of these T cells will start secreting factors which will attract more T cells to reach the tumor area, and then finally can then result in the cell death. I would like you to focus for a moment on the final process, the killing of the cells. We know that the stimulation factors include interferon gamma and the T cell granule content. The important one I'm going to be talking about is the inhibitory ones, what we now know as PD1, PDL1 interaction. So we'll see this in a little detail little later, but this is where we are having a large amount of developments in cancer immunotherapy. So this is a three-model system for humoral immunity. So we have the antigen presenting cells through the MHC complex. It can stimulate the T helper cells through interleukin 1. The T helper cell in turn activates B cell through interleukin 4, and this can mature to plasma cells and you can get your antibodies. We also have the three-cell model for the cellular immunity, and here we have the antigen presenting cell stimulating the T helper cell. The T helper cell in turn secretes interleukin 2, which promotes the sensitizing of the cytotoxic T cell, which then goes and destroys the cancer cells. Now, this is a very important diagram, and at the same time pretty complex, but don't worry about it. We'll try to break this down as simple as possible. So we'll start from here. So this is a normal cell within our body. And as I said in the previous presentation, we are exposed to a wide range of carcinogens, and these include physical, chemical, biological, etc. So once these carcinogens act, the normal cell can develop mutations, like either in the P53 or RB, which are tumor suppressor genes, or RAS gene, which is a proto-oncogene, where it becomes an oncogene. As a result of this, you now start seeing cancer cells emerging. So these are the red ones on the background of the normal cells. These cancer cells again exhibit tumor-associated antigens. Now, because we have what we call the cancer immunosurveillance, which we saw a little while earlier, these are then detected, and we have the entire immune system coming and fighting these initial cancer cells. So you have the natural killer cell, the natural killer T cells, we have the macrophages, the T helper cells, cytotoxic T cells, and the gamma delta T cells. So, and then we also will have some antibodies coming. All of these will start destroying the cancer cells, and as a result, the cancer cells are eliminated and you are now protected. I did mention this in the first presentation. Every one of us every day have millions of cell division happening, which is required to replenish or replace the skin cells, the intestinal epithelial cells, etc. Now, therefore, at any point of time, we can always have some of these cell divisions going wrong. And when that happens, the DNA repair mechanism comes into play. If the DNA repair mechanism also fails, then the cell has a potential to become malignant. And that is what is important. So we need to understand that the immune system plays a very, very important role in protecting us. Now, let's presume that the most of the immune system has destroyed most of the cancer cells, but the cancer here, for example, in this red one here, goes into a dormant state, and in the dormant state, it changes some of its features, and that's what happens. So you are starting to see what we call as the clonal evolution. So the immune system is producing changes which the cancer is trying to avoid, and thereby developing newer and newer mutations to survive the onslaught from the immune system. This process will finally result in a cancer development wherein they are no longer going to be under control of the immune system. And this phase is called the equilibrium phase, and finally it then breaks out where the tumor cells rapidly proliferate, overwhelming the immune system and inhibiting the immune system, and that is what happens here. So there is a escape from the immune system, and the immune system is no longer effective in controlling the cancer. So the main defense mechanisms of the tumor against immunity is alteration in the MHC class 1 and tumor antigen expression, and this happens by down regulation, meaning that they do not express these molecules. So if it is not expressed, the immune system cannot see, it's blind. So therefore they can survive. The tumor cells can survive. Lack of the co-stimulatory molecules, again the loop required for the signal generation is lost, and therefore the cells, T cells, become ineffective. And the similar factor, the T cell signal transduction molecules can also interfere, like cell death receptor signaling gets compromised. We also have immunosuppressive cytokines. Now, as I mentioned earlier, cytokines are chemicals which are released by a wide range of cells, including immune cells, even the tumor cells. It's one which can, some of these factors like the TGF beta, tumor growth factor beta, is actually immunosuppressive. We also have induction of immunosuppressive cells. These are cells which can inhibit the immune response. We'll see that in detail shortly. And secretion of a wide range of suppressive factors as well. So among the inhibitory cells or factors, I mentioned about the T regulatory cells. Now, T regulatory cells have a normal physiological function to prevent the immune system becoming overactive. So this is like a break to prevent the immune system from becoming overactive. However, in the context of cancer development, the cancers can co-opt these T regulatory cells, and as a result, the normal immune response cells like T helper cells, the T cytotoxic cells, or the dendritic cells cannot function effectively when there is a high number of T regulatory cells. Another important component here is the myeloid derived suppressor cells. Now, these suppressor cells can inhibit wide range of cells, including the B cells, the T cells, cytotoxic T cells, and also the dendritic cells and NK cells also. So all the positive ones can be inhibited. In addition, it also stimulates T regulatory cell production. There is a massive increase in T regulatory cell production, which again go to inhibit, as already seen, the T cells and the dendritic cells, etc. We also have B regulatory cells being induced. We also have suppressive dendritic cells coming into play and suppressive macrophages, what are called as tumor associated macrophages, coming into play. So as a result of all this, through wide range of cytokines or molecules like IL-10, for example, here, which is inhibitory to dendritic cells, or here you take the indoleamine dioxygenase, which depletes the tryptophan, an essential amino acid, which is required by T cells. So which means the T cells cannot function properly. Similarly, arginase enzyme can be overexpressed, and this depletes L-arginine availability, which again interferes with T cell function. So these cells, the suppressor cells, play a very important role in allowing the cancer cell to develop further. So this is a very crowded slide depicting the tumor microenvironment. So what I mean by tumor microenvironment is that in the tumors, you will not only have the tumor cells, but you will have these fibroblasts. These are called as cancer associated fibroblasts. You will also have immune cells which are trying to fight the cancers. But unfortunately, there will also be within these immune cells some pro-tumor immune cells. For example, here you find that the myeloid derived suppressor cell, which I mentioned a little while earlier, is here, which will now start interfering with the normal immune system function. We also have the M2 tumor associated macrophages, for example, here, and this is over here, which again interferes with the regulatory or the regulation of the immune system. And we also have the T regulatory cells, plenty of them moving around, which will inhibit the normal T helper and T cytotoxic and the dendritic cell function. So on the one hand, we have the anti-tumor cells, and the other hand, we have the pro-tumor cells. In addition to the cells, we also have what we call the extracellular tumor matrix stiffness. Some of the tumors which have high content of this stroma elements, the fibrous elements, include the breast tumor, pancreatic tumors, which will actually interfere with these cells getting into the tumor area, so that they cannot actually bring about destruction of the tumor cells. So this is again to tell you about the M1 tumor associated macrophage, which is protective, in contrast to the M2 tumor associated macrophage, which is inhibitory of the cellular immune system. And the myeloid derived suppressor cells actually again promote the tumors and mediate more immunosuppression as well. And interestingly, we also have the neutrophils, which are tumor associated ones, pro-tumor, which can secrete this matrix metalloproteinases, which are actually enzymes which break down that extracellular matrix, allowing the tumor cell to migrate, invade the neighboring tissues, and also spread to distant organs. And interleukin 1 beta can cause the natural killer cell inhibition. It inhibits its function. And this is where I mentioned about the PD1 and PDL1, which we saw a little while earlier. PD-1 is expressed in the T cell and PDL-1 is expressed in the tumor cell, and some of the tumors will have high levels of this PDL-1. Binding of PD-1 and PD with PDL-1 will result in the T cells becoming ineffective. The cytotoxic T cells cannot function adequately in spite of recognizing it. It will not kill this cell. So for therapy, now we have actually developed monoclonals either against the PD-1 or against the PDL-2. So these antibodies, here PD-1 and then PDL-1 targeted, will prevent this interaction between PD-1 and PDL-1 and thereby allow the cytotoxic T cell to destroy the tumor cell. So this is something which we'll talk about in another different presentation. These are the different hallmarks of cancer. One of the critical hallmarks is avoiding immune destruction, which is what we saw just now. And it is here we are now able to target a wide range of monoclonals. For example, anti-CTLA4, ipilimumab is a monoclonal antibody against this CTLA4, which is used in the treatment of cancers. So we find that now a lot of developments have happened in the context of developing newer immunotherapeutic systems and molecules. So I think I should stop here because I'd like to don't want to overload you with too much of information. I would like to thank all the large number of the cartoons and photographs, diagrams have been taken from different sources which have all been acknowledged. They were all fantastic figures, and I really need to thank them, which allowed a better presentation to be made. So I'd like to again thank you, and hopefully the next presentation should be on cancer immunotherapy, and we'll see what are all the newer developments which has happened in this context. I would like to again have feedbacks from you, and if you think there is some way I could improve, please let me know, and thank you again for your attention.